Thanks everyone for joining. I have the pleasure to introduce Dr. Jenny Van Eyck, who's a professor of medicine at Cedars-Sinai Medical Center in California. Also the director of basic science research in the Barbara Streisand Women's Heart Center and director of the new Advanced Clinical Biosystem Institute, where she recently moved from Johns Hopkins University. More recently, she's become the co-director of Cedars-Sinai Precision Health, focused on in-hospital population individualized healthcare. Jenny is an international leader when it comes to clinical proteomics, and her lab's focused on developing technical pipelines for de novo discovery of larger scale quantitative mass spectrometry methods, which I think is very relevant for this audience here today. This includes multiple reaction monitoring, MRM or SRM for those in the audience. And she's more recently driven some methods in the data independent acquisition space as well. Her laboratory is well known for a lot of very technical quality data generated results with rigorous control for tight percent CV, which I think we all strive for in any method we develop, and applying these to key clinical questions to drive progress in how we treat people with medicine in the end. The aim is to maximize throughput reproducibility of the methods that they develop, to move targeted and robust discovery of those methods to the large population so that we can all benefit and really continue that assessment for clinical grade assays to drive progress in the brain and cardiovascular space. And so some interesting facts about Jenny. She's not wearing her moving clothes today, but she's a moving lab. So there are 12 mass spectrometers strategically moving in the background for her today. I guess today's the wet lab move is what she offered to me. Jenny grew up and spent some time in a place called Cameron Falls, Canada, which I have yet to Google, but it's described as a ghost town. And so I definitely will go and look for where Cameron Falls is on the map. And her high school in Caparol, Canada, it no longer is there, but I'm sure that was a very good place to learn based on how successful Jenny's been. My own personal story for how I crossed paths with Jenny a few years ago, we crossed at a technical conference where she gave a presentation that really changed my outlook on statins, for all things, and some stigmas and how to approach those for your own personal health. So I still remember that to date, and I really enjoyed it, and I look forward to hearing from Jenny today. So Jenny, the floor is yours.
Thank you so much. I'm absolutely delighted to be here and honored to have been asked to give this keynote. And I'm hoping we can have a conversation about some of the things that we've been thinking about in our lab, and really based coming from the ability to be so analytically precise in some of our larger workflows or larger kind of more encompassing proteomics workflows, that begs the question of what should actually end up in the clinical labs. And can we be thinking about a paradigm shift that's coming at the same time that we've been making a parallel shift in remote sampling and the ability for patients or any individual to take their own blood sample anywhere, mail it to us or essentially send it and to be able to be then analyzed with the same precision as a clinical chemistry lab. So these two kind of things are going on simultaneously, and it opens up a lot of questions or potential solutions for precision medicine. So that's what I'd like to talk about today and get questions around that and what people are thinking. So right now, the way I look at it in the field of precision medicine is there's actually two things we're going to talk about. This is the first one, is that we really need to grow new approaches to precision medicine. We need to move from where we've been thinking to maybe a new way of approaching it. And I think the second aspect is that the breadth, I spelled that wrong, not breath as in breathing, but supposed to be breadth as in amount, and the accuracy of biochemical assays always matter. What you're measuring has to mean something, and there needs to be an output of what you're going to do, even if that output is understanding or output as in there's a therapeutic approach differences. And then finally, telehealth, I think, is coming on board, maybe moving a little bit faster now because of COVID-19. And the question of how does one integrate telehealth into medicine, and could this actually help to allow people to have their own control of their clinical chemistry results, the biochemistry, who they are, their results, in a different way than we've been imagining it. And I think with COVID-19 and this move to being able to adopt telehealth into the healthcare system may now be quicker than what we thought originally would happen. So the first thing I would like everyone to think about is really a paradigm shift in personalized medicine. So in the past, we used to have a drug and we would say, find me patients. We want to know who are the patients that are going to take this drug. And the whole concept between precision health or personalized health is the concept is we have a patient and now we need to find the treatment. So what that means for biomarkers is we go from a paradigm of subclassification to a paradigm of individual. And you may think that this is almost the same thing, but it's actually different in how you find those diagnostic markers or how you think about them. So you often hear in precision medicine is we're going to test everyone and then find that there's a pathway in people that we classify as having rheumatoid arthritis, for example, are similar to a class of people in heart disease. And, or we have a drug and we only want to be able to give it to a subset that are going to be responsive. That's subclassification. An individual, at least the way I think about it, means that we have to understand you, all of the diseases and physiological processes happening to you, and then predict how and what drug should be used or what therapy should be used for you. And that therapy can be health related, or it could actually be an intervention around a drug or a drug treatment. And that also starts to have implementations or implications in wanting to measure people's changes over time, right? Because we now want to look at you as opposed to single point in time and subclassification. So these are two very different paradigms about how to think about diagnostics and prognostic markers. Neither's wrong, they're just very different. And when people talk about precision health, they don't usually distinguish between the two different possibilities. So this is what we want to be able to do. We want to be able to find those biomarkers that will change how Beth is going to be treated for breast cancer, for example. With breast cancer, she'll go through possibly multiple different drug treatments as they change it to try to find the right one to fit for her. In cardiovascular, the same thing with statins. If you're not doing well on one, they'll change it until they find the right dose and the right make for you that's going to be most effective. So the question also becomes, could we find a diagnostic to make that faster? And then the second question, which we're going to end up in at the very end to kind of give you some tantalizing dreams that we have, is to ask the question, can we actually have a surrogate cell model or organoid model that represents Beth, comes from Beth, that we can actually do the drug screening on them before we actually move and make the decision of which drug would be best for Beth. So we want to be able to move from the concept of an individual having and treating one disease, but more of a holistic, that they are a combination of who they are, the genetics and the proteomics and the metabolomics, the environmental impact of what's going on with them, their past health status. We want to move from the concept of having a single biomarker to a more ability to monitor mechanistic networks. And we want to move from one time point to longitudinal time points. And finally, we want to combine all this and move into telehealth and really move to what might become a next generation of medical practice. So this is the stuff that we're going to talk about for the majority of today's session. It is in its infancy. It's a concept, and of course that means it will change over time as we develop the technology and the platforms required and start to launch these concepts into to the broader world. None of these are unique to our lab. Of course, lots of people think about moving from one disease to a holistic approach. Many people talk about more having multiplex markers. So we're just combining this together to come in and say, how can we practically move into a healthcare system and change it? So let's start with one marker to a mechanistic network. And from a discovery point of view or from an epidemiological point of view, this makes sense to do. But does it actually make sense to do in the clinical lab? And how would one do that? Because there's a lot of analytical work that needs to be done in any assay that goes into the clinic. But let's go through the logic first and why we want to do this. So the premise behind it is that diseases, even a single disease, is not represented by one sentinel protein, but rather the disease is due to a network of interconnected proteins. And the closer you're to that interconnected network, the better you are going to be at being able to understand its impact as you move towards treatment or as the disease is developed. There's also an implication underneath that says that often diseases aren't a single perturbation. It's not a single pathway but actually a whole bunch of them, unless you have a genetic disease. But if you're talking about complex diseases like cardiovascular disease, it's not a single disease, but it's a spectrum of diseases or mechanisms that can come out and give you that clinical phenotype. Then the second premise is that at each disease stage, as you progress, there can be multiple underlying processes. That's what I mean. It's not, you have cardiovascular, you also can have diabetes, you can have different kinds of diabetes. You could have been exposed to poor nutrients when you were young, so you have an added risk. So there's a lot of things that go into it that makes your disease yours. And understanding how you're going to respond to therapy or what state you're at actually probably has implications across multiple processes. The third concept behind it is that these differential combinations of networks should better define you as a person, your health status, how you'll respond to a drug, as opposed to a single one. Finally, we know that protein networks are dynamic, and this ability for proteins to represent a huge chemical space because of post-translation modifications, protein isoforms, these huge networks should allow us to be able to differentiate you better from another person, your health status, but also because it's dynamic and things change and your proteins change in response to your environment and the perturbations that happen to you on a day-to-day life. This will also allow us to better pinpoint with time and enough knowledge. So what there really needs to be able to have is the ability to monitor these multiple networks to find out which of those are going to be the best differentiators and really deconvolute these networks and match them over to their biological processes. And that's what I'm trying to illustrate on the right-hand side, going from a single marker to these different networks. So for example, if you have CRP, a great protein for measuring inflammation status, to having in our hands, these are two of our targeted assays. One is an acute phase protein assay. This is an IMRM assay, so immuno-enriched peptides 2 spectrometry. It is from Ciscapa, a company, versus our health surveillance marker, which is a larger marker composed of 54 proteins. So the 3 assays we're going to look at is the acute phase, 11 proteins representing the acute phase response. So acute phase response is more than just CRP. It's a cluster of proteins that are in response. They change with time and change differentially depending on what is stimulating the acute phase response. So this would be, for example, a single network, even though there's multiple prongs in that single network. A health surveillance is comprised of 52 proteins, and those 52 proteins represent 9 pathological processes: innate immunity, vascular response, platelet activation, heart, lung, kidney function. It is a combination of more holistic 9 pathways. And if you monitor those 52 proteins, you can see in different individuals, as they get treated, a differential response depending on what's going on. So it's different pathways. And then finally, we have discovery, which is often cited to be where you start on the process of finding biomarkers. You try to look at everything as much as possible, and then move as you go towards clinic to smaller and smaller number of proteins. Part of that is logistics, part of that is cost, part of that is the analytics around the precision of the final biomarkers. In the clinical chemistry lab, you really have to have a 20% coefficient of variance on any protein you're doing. And all of a sudden in our hands, as we're using these discovery technologies, they've become so reproducible that we're hitting a 20% CV for the majority of the proteins that we are now undertaking in these discovery efforts. And I'm going to go through that data to show you that. And if that's true, it begs the question, can you use these broader based assays as your single clinical assay and only take out those sub-networks that you need to do to give that information to a patient? So that's a very different concept. That means instead of having a whole slew of mass spectrometry assays, like the acute phase, like the health surveillance, you would have a single assay that all of these proteins are so well done that you can then move those assays and report only a subset of those proteins clinically. Now, in genomics, they are doing this, or at least there's a lot of talk about doing whole genome sequencing and then only releasing a subset of the data on the clinic. It's not there, but there is a possibility to do that. So this is no different in conceptual ideas than doing a whole proteome on someone and then releasing a subclinical. There is so much behind that on the analytical side we have to deal with. There's a whole bunch around the cost and benefit around doing something like that. But it does open up the possibility that we can start to think about having these networks that perhaps can find that balance financially between cost and benefit in certain aspects like long-term monitoring or therapeutic response, where you can have impact of having a cold or a flu to find out whether or not that's what's happening, or are you actually rejecting your heart, for example. So in the concept of moving from discovery to a clinical use, this is the pathway. You go from targeted to pathway specific to a single protein representing that pathway. This is very classical, from lots of proteins to very analytically precise single proteins, be that a mass spectrometry assay or an immunoassay. So going from DIA mass spectrometry to something like an immunoassay around CRP. So this is where our assays sit. We have DIA discovery, holistic, trying to see as much as we can, moving to something like the acute phase immuno-MRM assay, moving to something like a CRP assay, right? And what moves you along this is really the analytics between reproducibility, precision, and linear response, performance criteria. This is what moves you along the pathway. So now, if all of a sudden I say, I can make the DIA mass spectrometry discovery equal to, on the analytics, to a CRP, what does that mean? Of course, it doesn't mean it's going to happen today. There's lots of things, as I said, that stops that, but it does open up the possibility of shifting, certainly in academic centers or in certain clinical conditions where you need long-term monitoring, where it's a super complicated case to be able to do it. So let's take it. So behind our DIA mass spectrometry, we have a lot of QC issues, a lot of QC of when we need to redo samples, everything from sample processing to the MS. This is just one example. We do about 10% QC samples and a 10% redo rate on our current DIA mass spectrometry workflows that I'll go over to you. So the way I think about the discovery versus a clinical assay is that we need the same performance. The same concern at every step is also there at DIA mass spec at the discovery. It's just maybe not quite as tight, but we need to strive to make it just as accurate and just as precise and just as reproducible as much as possible. And so this I'm going to talk about is actually from our COVID-19 academic team. I'll be talking about them throughout. They revised our whole DIA workflow, and I'm very proud of them. It was led by Angie, and there's their name. So this is to show you kind of where we are. It's going to get you kind of thing. So gradient time, the amount of time on the mass spec, number of proteins that we're quantifying, and the size of the dot is the CV. So for example, you can see the acute phase IMRM. It's 11 proteins. It's a black dot at the bottom. It's a coefficient of variance around 3 to 5% on those proteins. It's a clinical assay. That's where it needs to live. Our health surveillance, it's 52 proteins. It has a coefficient of variance on average around 13%, so still doing very good. Right now it's a 60-minute gradient. We're moving it to a 30-minute gradient. And you can see that those are targeted assays. In the green are the discovery assays. So we're going to talk about 3 different ones. We have our ultra-fast one. It's a 21-minute gradient. It's a 25-minute turnaround time. It's an evocept on an Exploris. So it's not on a triple quad. It's a DIA mass spectrometry. And you can see the size. We've cut out to 40% CV. And you can see the little green dot, that's the total. So the total of proteins we quantify is around 250. I'll show you the data. But our CVs for most of those proteins are under 40%. And then we have 2 deep profilers. This is when we do depletion, is profiler 1. So we do both depletion on a 21-minute gradient and naive on a 21-minute gradient. We'll talk about that. So every sample gets run twice. We get a large number, and you can see again. And then the deep profiler 2 is a 60-minute gradient, so about a 75-minute turnaround time on the mass spectrometer. And you can see how we're doing it. We're moving from a targeted on triple quad to an Exploris using either Phenomics column or an evocept system. And we'll go through that. And this is based on a 3x3x3 reproducibility. So each sample prepared in triplicate per day, run in triplicate on the mass spec. So each sample done 9 times. on 3 days in a row. So this is to show you the first one. So this is looking at the ultra-fast 21-minute gradient versus a 60-minute gradient. So 60-minute gradients are a typical one that we would load it on. This is under optimized conditions for both of them. And what you can see is mean intensity up the y-axis and CV along the x-axis. So Y is mean intensity to give you the breadth, the depth of what we're covering across the coefficient on a 3x3x3 reproducibility study. And you can see that in dark blue, this is our coverage that we normally get when we're running a 60-minute naive sample on DI8. It's beautiful. And you can see the faster one, of course, isn't going to see as many proteins, but it overlaps. But what I want to point out is the dotted line here. This is your 20% CV. And you can see that, in fact, there's a lot of proteins under 20%. And then if you move to 40%, you're capturing a huge number of that proteome across the board. And the reason we can do this is because we have automated the whole pipeline. So we now have a module pipeline that has maximized the automation. So we can take whole blood, which is from the remote sampling, plasma or depleted. This is removing the top 15. It's also being automated. So it's on a 96-well plate. We do all of the automation system. And now we're actually also doing desalting on the automation system. This is the I7 by Beckman Coulter. Everything's been automated. And then once it goes on the mass spectrometry, we also have an automated search program. It's called OnOptra, and that's really coming from our bioinformatics team led by Vidya, who is quite remarkable. At every step, our data is backed up. It comes directly off the mass spec and backed up without any hands on. It goes straight to deep storage. And then we have different places that it's also backed up and we're not able to access. So we have both an automation on the sample prep and automation on our data analysis. And essentially right now, you get your sample, you put into the 96-well plate, it comes off the back end and it's ready to go on the mass spec. So it's all hands off, and then it goes off the data analysis, and we have our QC samples being run daily so we can know what's going on. So it's been working very well. So one of the other things we worry about is actually linearity of the response. And this is looking again at the 60-minute gradient, the traditional one. You can see this is just an average across. You can see it's a very beautiful linear intensity over as we dilute it out. And of course that matters which proteins. As you go to lower intensity, of course, that also changes. But this is overall, most of the proteins are very good. But you can see on the EvoSep system, on the 21-minute, you do have a much tighter sweet spot of where you have to hit your linearity your load in order to get your linear. Much more sensitive to load in order to make sure you're in your linear range. You can see that right here on how tight it is. Still linear, but it's a very much smaller linearity. But if you do it, then you can get a linear response, and you can essentially do 60 runs per day on a 21-minute gradient. That's a remarkable increase in throughput compared to a 60-minute gradient where we're running around 18 samples, just 20 samples per day. So going from 20 to 60 is a huge jump in throughput, but of course at a cost of whether or not you're capturing not just the proteins you want to, but more the biological processes. And that's what's shown on the right-hand side. So down, you can see different enriched biological processes that are captured by the proteome that we're seeing in plasma. And you can see as you look for the naive 21-minute gradient, these numbers here represent the number of proteins in each of those networks for the 21-minute versus 60 minutes. So of course, the 60-minute, you're seeing more proteins. It's a bigger gradient. You're going to get it. So for neutrophil degranulation, you get a great representation of that network in the 21-minute gradient, but it's 21 proteins. When you go to the 60 minutes, you have 134 proteins representing that process. Now, do you need 134 proteins to represent that process? Maybe not, but you do capture additional processes in the longer gradient. So depending on your question and what you need to ask, you can decide whether or not you want to do 60 runs per day, or do you want to do 20 runs per day, and at both a differential in cost as well as in coverage and throughput. So for example, if you care about angiogenesis, which I often do, it's a great, or the VEGF signaling system, which is again a very important signaling system, you might want to do the 60-minute gradient. So the question is, we came up with it, is there a compromise between these two circumstances? Could we make the advantage of a 21-minute gradient and do it, so we capture that in a 21-minute, but then do a second depletion, because take the same sample, again, it's only 10 microliters, so it'd be 20 microliters in total for each sample. 10 gets processed for the naive, and 10 gets depleted of the top abundant proteins. Now, lots of people are skeptical about the depletion process with respect to reproducibility. And I would say for us, we've been working on it for a long time, but I'd say there's been a breakthrough, both with the advent of camel antibodies, so single chain antibodies to the top abundant proteins, and then us moving it to the 96-well format. And we've been able to get quite good reproducibility. It adds, generally speaking, about a 10% increase in CV. So of course, it's not going to be as tight as if you didn't do the depletion, but we're getting very good reproducibility still. And that has allowed us to now take samples and do a 21-minute gradient on naive, and then a second run, a 21-minute gradient, which on the depleted sample, which makes it to be a 42-minute gradient. So it's in between the two, and we found that to be very helpful. And so this is showing you on the left-hand side the linear range for the depleted. This is a 5 by 3 by 3 reproducibility using a 21-minute gradient using the depleted sample. And you can see it's very good. And the numbers in the CVs actually are just, we were so excited. And on the right-hand side, you can see how the coverage of the pathways are now quite good between this dual workflow where we do naive and the depleted on the automated system. And you can see we're getting better coverage now of even additional pathways of interest across the board compared to even naive situations. So again, depending on what you're doing and your throughput and your cost, you can now have a decision of which protocol you want to use. And all of it's done on the same automation system. And you essentially put your sample on, press your button, comes out the back end, and you can put it on your mass spec. So it's been really quite remarkable. So this was to represent the ripple effect. So how you can do it. So as we know, as I mentioned earlier, in the genome world, we're moving to a concept that potentially we want to be able to do whole genome sequencing and release a subset. So are those workflows good enough to start to think about moving a conceptual idea that we would be using large screening of individuals in order to be able to give a better clinical decision? And when would you do that? So if you already have a heart attack or you think you have a heart attack, you're not going to move from running cardiac TNI. That is a great marker. You're going to run TNI to diagnose having a heart attack. But there are situations where complex diseases are essentially that they're complicated, or the response to drug is complicated, and understanding more could have great value, potentially. So that's the question we have to think about. And the other thing we have to think about is, can we understand these networks, especially if we're doing it over time? And so if you can be that accurate, and if you can measure these individuals over time, how would that impact the decision that we're making today in the clinical chemistry lab and in medicine? So how can you think about that? Well, one way we're thinking about it, as many other people, is in the concept of remote sampling devices, where we can go on and actually take a pinprick of your own blood and send it. So this is a Neoterix device. There are other devices out there. This is a 10 microliter one that we use. You essentially pinprick your finger, wick up 10 microliters, and then mail it to us using US Postal Service. And then it comes into our lab. We can digest it on our automation system and run our whole blood protocol. So this is what the old kit, they just revamped this kit for COVID-19. So it's physically smaller, but you get your kit, you have your instructions, there's a video, you have your devices in a clamshell. This is the 4 devices, you can get 4 or 2. You get your bandages, your lances, and then you have this little bag that you would stick your clamshell once it's filled into. It has a desiccant, it's foil, and then you have a self-addressed envelope, you stick it in there, and you mail it to us. Dried blood, once it's dried, is no longer considered biohazard, and so it's protected in this foil bag, and it can be mailed or dropped off. Of course, there's a whole, I don't have 7 cents here, but there's a whole world of great companies working in this space. And I think that's wonderful. I think we need to be there. We've done releases where we do not train people. We just send them the kit and they're able to do it on their own without having any face-to-face contact, which I think is also very important. That is with Dr. Barry Mers in our Takoshiba women's cohort. So I'm going to give you a little bit of actually our first one that we did, and this was funded by the California Initiative to Advance Precision Medicine, where we wanted to be able to predict major adverse cardiac events. So having heart attacks, sudden death, arrhythmias, or rehospitalization due to heart failure. This was in what we term to be mid-risk cardiovascular individuals. They have about a 10 to 20% mortality in a year. That's mid-risk. If you're high risk, you're at a higher risk than that. So of course, there's great value in trying to understand who is at risk when, and you want to be able to monitor them quite quickly, right? Not just once a year. You want to be able to do it. So this was our first one. We linked up with Brennan Spiegel as well as Dr. Barry Mers, where we also had Fitbit data coming in. And we also looked at the traditional markers of brain natriuretic peptides, the BNP. It's a great looking at your blood pressure, cardiac troponin I, and the apolipoproteins. We did that by a targeted mass spectrometry assay. So let's talk about the device. So this was our first time. How it worked is you got trained originally, and then you would take your kits home with you. We emailed you, the day before and saying, hey, 1 month later, take your blood sample. Don't forget to mail it to us. And they would then send it. And this is a number response. We actually had 198 out of the 200 people agree to do it. And you can see that we actually had really good compliance. The two blue are the number of people who mailed it to us. And then on our exit was on day 90. So it was a relatively small study. And over here, you can see the number of responders over the weeks. And the dotted black line, this is Mitra, the device, the remote sampling devices versus Fitbit. So we had incredibly good compliance in this program. And we remain in all of our studies, which we have a lot, to have that kind of compliance. But there's a difference between compliance and filling your tips perfectly. So this is what we learned here. So you're supposed to fill your tips like this. So they are 10 microliters. This is the advantage of this technology. But of course, we got differences. So what we learned from that very quickly after about a month is that we had to improve the instructions, which we have. And I can tell you now that this is not what we get. We normally get about 90% looking perfect. So we learned fast. And now with better instructions and better videos, we actually get really not just good compliance, but we also get good quality tips. And we always do 2 tips per person. So usually one of them is always excellent. And again, we work on that. So again, it's not just compliance and response, it's the quality of the tips, of course. And that's true for any of the remote sampling devices having it. And we did a lot of work behind the scenes originally to understand the stability of these devices at room temperature when it's hot. If you're sending that sample from Arizona and it's sitting on the tarmac at 120 degrees, you need to know that whether or not that will affect your sample. So all of that has been done. We know, generally speaking, that we want to have the sample to our lab within several months, about 3 to 4 months. And once we have it, we put it into -80, and it's good for at least a year. We haven't tested past that. We have incredible, because we're digesting in the tip itself, we put the tip into that automation system with buffer. It's all digested with the tip in there. We actually get incredible extraction recovery, and we get very good digestion efficiency. That's all been optimized. So again, it goes through the same process as plasma with high reproducibility. And this is what it looks like. So this is a 3x3 study again, mean intensity over mean CV. You've seen already the plasma, that's in blue. That's a 60-minute plasma. And now you can see in red the coverage that we're getting with the blood proteome. And again, 20% CV, we're getting really great coverage and very great reproducibility. This is the data, the cumulative frequency on this of coverage of the proteome that's hitting 20% CV. And you can see at 20% CV on a intraday, we're getting incredible recovery. On intraday, you're really at 40% is where you want to be able to get 80% of the proteome coverage. So 80% of the proteins that we're quantifying on dried blood have CVs with intraday variability of 40%. That's phenomenal. And with improvement, we think, of course, we can get that down to intraday variability. And so we just need to move this guy back up. So that's a day-to-day variability. We need to be able to improve that. But you can see, even so, 80% or 85% of the proteome being quantified in plasma using DIA mass spectrometry have the CVs that are reaching a point where you can start talking about using it really as a high throughput accurate assay. So this is one of the things that is on the back end. You don't actually have to know what this is. This paper has been published, but this is the analytics. You know, it all looks great on the mass spec, but you're not getting a buttload of data. And we have to be able to, again, be very careful and ensure that the quantitation is accurate and precise. And this was the algorithm that we published around just 11 apolipoprotein panel. It was on the 199 individuals that we were talking about over the 4 time points. So we had 741 time points. Samples were done in single. We tried to get most of them as duplicate, but we did not all of them because some of the, as you saw, those tips were not good. So we analyzed just over 1,100 samples for the apolipoproteins, and we had 2 peptides per protein, 3 transitions, and we had internal standards. So we actually, for that project, just to measure 11 proteins, 199 people over 4 time points, 72,000 chromatography peaks to look at. Now you know why we have to have a really good algorithm that helps us sort that through. If it's clinical chemistry, we would look at each single one. In fact, we did that here. And so we made the pipeline that allows us to be able to do runs, and our philosophy is now published. So where could we use this? Where would you think about using this? Because it is it's a lot. So one place that we're using it or starting to use it is in the Center for Undiagnosed Patients here at Cedars. So this is, we call it the CUP, Center for Undiagnosed Patients. It is a team, this is just a phenomenal thing. So if you are a complicated adult or a pediatric and your clinicians can't figure out what's wrong, you can apply to become part of a patient in the CUP program. You will meet with 5 physicians who represent different diverse specialties, and that is decided who you see depends on your state. You then meet with those individuals, and then your report goes to 20 clinicians, and there's a brainstorming about what to do, and they decide on your additional diagnosis, different tests, and then eventually we hope to be able to either rule out diseases or rule in a diagnosis. And so the concept behind it is that people who are clinically complicated could represent new syndromes, rare genetic diseases, or complex diseases, or known diseases, but have rare clinical presentation. So we're now starting to do proteomics on their blood samples over time, 3 time points per patient, to try to help understand in these complicated individuals what might be going. And as you go through treatment and therapy, we'll be able to look at responses and find it. So this is a place where you could see having a broader view of what's going on may help, may not. We'll find out. This is one of the projects that we're doing to try to understand undiagnosed patients. And anyone who knows me knows that my oldest daughter was undiagnosed for several years. And I can tell you the day you get a diagnosis, your world changes and you have something to understand. So very important. So we want to be able to add value outside of that in the context of telemedicine. So right now, you go to your healthcare facility and then they send your samples to the clinical lab. And then the data goes back to the healthcare and then you get it. So you need to go everywhere. So we want to move to it so that you can get your own sample done at home or wherever you are and then send it to us. So we changed the whole paradigm. We're moving and shifting how the cost benefit, not just to the healthcare system, but to the actual individual that's interacting with the healthcare system. So the Choral study in COVID-19, I'm just going to give you a few examples because we're running out of time around what we're doing. So our hypothesis is that, even those individuals who are positive or asymptomatic or have moderate clinical presentation and may not need hospitalization, even them will develop multi-organ dysfunction over time. I think this is not, this is not novel to us, this is coming out, and I think more and more people. So with Susan Chen, we have something called the Choral Studies. It's a whole bunch of cohorts within Cedars-Sinai. And I want to just tell you the story behind the title, why it's called the Choral. So it's a Coronavirus Risk Association, a longitudinal evaluation study. And Choral represents, and I remember this day, on March 14th, 2020, when quarantined Italian citizens started to sing from the balcony. I think everyone saw those videos. So we were so inspired, we named our studies Choral. This is one study. This is a longitudinal frontline nurses. This is with Susan and also Margot Mission. This is her over here. She's an amazing PhD nurse. And it is a mega kit. We send home the Mitra devices. We also send home Tazel devices where we're doing monitoring the acute phase, discovery, metabolomics, looking at seroconversion for frontline workers. And they take their own samples weekly. And we're monitoring them for 6 weeks. We have the baseline healthcare workers. This is everyone at Cedars could participate and get their looking for IgG conversion. And they also had to donate a second tube of blood for research. And we have around 6,000 individuals in this cohort, and we're now expanding it to one of the other hospitals. We expect to get up around 10,000 individuals. And a subset of them, we have both the Mitra devices and plasma. So it will be the first time we have that many individuals where we have both sample types. And that's me getting my sample done. We had thousands of people per day coming in to donate their blood sample. And I can only say, not only to everyone who was involved in setting the study up, it was a 2-week setup period, can you imagine? And everyone who participated, this is just phenomenal resource for us. And you can see we have a 4.8% anti-COVID IgG positivity over 6,000 individuals. So it's actually 6,318 individuals. We're now moving that number up higher. And we're also doing genomics on a subset of those individuals. This is the first DIA. This is the comparison between individuals across time. Doesn't mean anything, just to show you we have lots of beautiful data. This is using DIA on the 21-minute gradient, and already you're starting to see subsets of individuals, right? That's just, it means nothing, but it's just to show you we can do it. So where are we? What's possible? Well, I think maybe we're getting there. What we talked about at the beginning, going from one disease to holistic, one marker to mechanistic networks, maybe moving from one time point to a longitudinal in specific area, and then exploiting it. So we are doing this in the concept of precision biomarker labs. These are our new labs. There's 3 labs under one umbrella, a proteomic contract research lab where anyone can contract us to run your samples, and we'll run it. It's your data, we run it for you. The other one is commercial development, where we'll take, if you think you have candidate biomarkers, we'll take it through do the clinical utility, set it up into FDA or as an LDT for you to be able to release it. And then finally, we have a CAP-CLIA certified clinical chemistry mass spectrometry. So you can go from the beginning all the way to the end. You can use any one of these services with the expertise going from discovery all the way into clinical application. So these are the 3 labs, the contract lab, the commercial development, and the clinical specialty lab. Corey Brystron is overseeing the translational lab and the CRO lab. And in our field, people know Corey and know how remarkable scientist and operation person he is. So we're very delighted to have him. So I'm just going to end with one concept. So if we can do this with diagnostics, if we can do all of this, that will be great. But you know what? That's really only half of it. So we need people to be working on this side, the ability to individualize drugs. And I'm going to tell you our concept in about 1 minute. So here at Cedars, we have a huge regenerative medicine program run by Clive Svensson, where we take human iPSC cells and we can use them and reprogram them into a whole host of different organoids. We can do it, take a woman's iPSC and drive them into breast tissue. We can drive them into a motor neuron. And if we can do that, we can maybe individualize your therapy. We don't have to do the drug treatment on you. Maybe, it's a big maybe, we can actually do the drug treatment on your iPSC-derived breast tissue or heart cells or whatever it is and you're doing. Let alone what we could do. And all of a sudden, we're starting to add biological variability into the question of drug selection. So to do that, we have to be able to move proteomics also into that. So we're now doing iPSCs on 96-well plates and being able to move them onto automation systems. So again, the idea is grow them, don't touch them, put them right on the automation system, and then put them right onto the mass spectrometer. If we can go to smaller well size, it's even better. More perturbations, higher throughput. And so we're doing it both on traditional instruments as well as using CE mass spectrometry. This is our CE data. I'll be talking about this later at a CESI annual meeting. It's coming up in October. We're able to get, when we do a fractionation, all on the tip on the automation system, around 2,000 proteins with really good CVs. Not as good as what I just showed you, but really very good. So we're getting about 2,000 proteins, being able to do them in a high throughput manner. So this is what we're working on now. And the idea is be able to move even from that to organ on a chip. This is an organ on a chip again. This is the blood-brain barrier where we've already been able to show that we can run, when you take iPSCs and drive them into motor neurons or into endothelial cells, you actually make a little vessel here. It's like a little capillary. You can pump blood. You can see the blood going through here. And we can measure molecules and proteins going from the blood into the little tissue up here. This is a little tissue. It's a little organoid. And here what we can do on the proteomics. So just on a plate, we can get lots of proteins, around 3,500. And on the chip, just taking this little piece of tissue, this little neuron patch, we can actually get close to the same number on the chip. So this is going really, really well. So that's what we think is the future, not just the diagnostic, but the therapeutic. So we want to be able to expand the physician's toolbox. We want to be able to interconnect the phenotype, the clinical with the proteomics and move ourselves forward. Can't do that without a huge number of people. This is them listed. This is in bold are the COVID-19 team who has done an amazing job. I'm very proud of them, as I am very happy to work with everybody here at Cedars and with all our collaborators. So thank you. I'm done.
Thank you, Jenny. It's really very revolutionary and inspiring talk. Thank you so much. I think we are going to move to a Q&A. We have some questions from the audience. The one with the highest vote is, there are two questions, right? So I will read it out. Thank you for the fantastic talk. I have two technical questions. First, how were the error bar calculated in the figures of peptide intensity versus CV in the first part?
Yeah, so this is a 3 by 3 by 3 assay. So what that means is we take the same sample and put them into 3 wells, and we do the sample prep on that in triplicate, and then they put those samples. Each one of those samples is put 3 times onto the mass spectrometer, and we do that for 3 days. So the CV and the intensity is the intensity, the peak area under each of those runs, right? So it's 9 runs per day. And then that also gives you your CV, right? And then, so that was actually your interday, so multiple day CV, and then the average intensity of all of those runs. So that gives you that axis.
Thank you. That sounds very familiar for the analytical work.
Yeah. It's no, it's like I said, it's not inventing anything new. What it is just using what you do in the chemical chemistry, move it down to the discovery side.
Yeah, definitely. That's the the way to do it in order to control the quality of data. The second question is, could you comment on the software/peptide library you are using for DIA data processing, and then what are the number of peptides and fragments to define as the threshold for processing?
Yeah, it's a great question. So I think library builds are probably the most contentious conversation we have in our lab. So for the plasma, we've been using the twin library that was developed by Rudy Abersell. We've developed our own libraries many times, and including ones where it's across all of our disease states that we work on. And we haven't found that to help, which is completely different than we're talking tissue or cells. With tissue or cells, we find having a sample-specific library is essential. And we go to great lengths to develop those cell-specific, tissue-specific libraries, and they help and improve. So we handle them differently, which is kind of weird, but and we've talked about taking all of our cell libraries and make that into like a mega library and then use that against plasma, which is kind of a similar concept. So I think plasma is still a bit of a mystery to me, how best to design it. We're still, like I said, trying different ways. We have a Parkinson's library that we just developed for a grant. It's both plasma, and we're building one for CSF for that project. So, but certainly for cells and tissue, it has to, as far as I'm concerned, it has to be cell specific. We get better quantitation, and I think maybe there's just more unique proteins being expressed, uniquely expressed in those cells. So that's what we do. We use, with respect to, I forget the other part of the question. There was two parts to that question. I can't remember the second part.
Yeah, the software.
So we have, so the best way is to actually contact me. So we have this, we use MapDA, we use OpenSWAS, we have a whole bunch of other pipelines. We have a pipeline platform, an architecture called Optra, where we have a whole bunch of open source software. And then we have pipelines that allow you to mix and match what you're doing. And then that's settled down. And so that allows our biologists just to keep it always the same, depending on what they do. And we also have libraries in that where we have specific post-translationally modified enriched libraries. So we just published it, just came out in JPR, where in tissue we can measure, we have a hypermethylated, hypercetylated, hypersuccinated library for tissue where you can run a normal DIA assay and just do protein quant, but you can also do it against these PTM enriched libraries and actually in the same data set pull out the enrichment of the methylation and succinylation and acetylation. There is a trick to that one. It's not quite that simple. So all of that's on this platform. So if you want more details, please contact me, email me. I'll introduce you to Vidya and we'll go through the whole pipeline. Hope that helped.
Thank you, Jenny. Doug, you want to go to the next one?
Yeah, thanks, Dot. Jenny, absolutely spectacular talk. Really enjoyed it. It was fascinating, and it's great to see the progress your group's driving there. I'm going to combine two questions that were sort of related. So can you tell us if there's any regulatory approved protein biomarkers which were discovered via proteomics?
Yeah, so certainly discovered. Yeah, so there are several in the area of cancer that are now being used clinically. In our own work, probably the ones that are furthest along are in the area of traumatic brain injury, which we discovered while we were at Hopkins, and it was licensed to a company called BrainBox Solution. It used to be called something else, now it's called, and they're in FDA trials right now. And there are was 6 markers that came out of that. Well, it was a bigger marker, but 6 that were in the final draw. But you actually only need 3 of the 6. So the final marker set is 3 proteins plus an algorithm that's based on clinical information for being able to prognose the first 24 hours, prognose, obviously, TBI, diagnose TBI, but also prognose the first 24 hours. So that's traumatic brain injury. It's based on cell-specific leakage of subclinical brain injury. That was the discovery cohort, and myself and Alan Everett did that work. So yeah, it's a very long process, and that's one reason we made the Precision Biomarker Lab. Is I think it takes too long to go from discovery to a clinical assay, and more importantly, the technology changes, the expertise changes along that line, and a lot of those biomarkers, it's hard to raise money around because it's discovery. It's still so loose. So we want to find a way that allows increase the probability of getting markers through, and to do it quicker. So instead of 10 years, we want to be able to do it in about 3 years, and that's a Precision Biomarker Lab. That's a translational lab.
So that's great, and well, it's good to know at the end. We wish it was easier. The second part to that question is the role of MS in that diagnostic lab for hospitals, right? And obviously, I think you see it growing, but then another question that's related to this is insurance companies and their ability to cover mass spec-based diagnostic tests. Where do we stand right now with that?
Yeah, so I think, for small molecules, it's been there, right? So the question isn't that can they do it? They do it already for small molecules. It's there all the time. Mass spectrometry is used routinely for small molecules. The difference is a peptide's a big small molecule, right? And that makes it more complicated to do than small molecule. And so then the question becomes not necessarily can you do it, but the value of the assay. And I always say it should not matter how you get the data. If the data is of the quality that's allowed to in the clinical chemistry, it doesn't matter if it's an immunoassay or a mass spectrometry assay. It has no relevance. It's the value of that which the HMOs and the clinicians need to see. It's not how you do it. It's the value of the assay. It always comes down to it. So I think the trick for mass spectrometry is finding those assays where the immunoassay, like what we did for the one I just talked about, it's actually an immunoassay, 3 immunoassays. We moved from mass spectrometry to an immunoassay because it has to be in the clinical chemistry lab in a very quick turnover. And mass spectrometry is not there. Where we have our mass spectrometry assays are in chronic disease, where turnaround times are different, right? So today, mass spectrometry, at least in our hands, when we're thinking about the process of the sample coming into the clinical chemistry lab, it has to be processed. It's just, we're not there yet. We're not good enough, at least in our lab. So we're looking at chronic diseases and the implementation there. And that's where remote sampling helps. And remote sampling also helps because you should, if it's done right, have more samples coming through the clinical chemistry lab. And that changes, again, the finances. If you're having 1 sample versus 6 samples, your costs can go down. And all of a sudden, the mass spectrometry assays become also into the same game part as what immunoassays are. So again, there's lots of stuff that's outside of just the analytics. But to me, you can't even talk about those things unless you have the analytical framework solid enough. And then you need to drive those assays and the clinical content.
You want to take the next one?
Yeah, there are technical questions. Actually, maybe I should combine these two. The first question is, can you describe your automated sample prep system? Is it one platform that does the depletion and digestion?
Yeah, so we developed it with SciEx and Beckman Coulter several years ago. The first publication is one of those mega publications. It's in Journal of Proteome Research, and then that was done on the NPX system, their original Beckman system, and now we have it on the I-Series, and that's in a Jove publication. You can get it, and it's also in other publications. Jove is very, very specific. It's written out as a manual, essentially, and that's why we did it. So it was set up to do everything, and so we had what we built first core, so that's protein denaturation, reduction, alkylation, digestion. Optimize the digestion for MRM assays. It's about a 30-minute digestion. For DIA, it's about a 60-minute, but of course, you have to figure out for yourself what that's going to be, depending on, always the digestion part. And we've added desalting. It's a positive pressure desalting system, so we have that also online. And we're just now moving the depletion onto it. So we've done it a lot manually on the 96-well plate, and we're just now putting it onto the automation system. So probably within a month, everything will be automated from depletion and all the way through. And of course, you can use that front-end depletion. That's a capture. You can use it for if you wanted to just isolate specific proteins. You have other antibodies there, right? So if you wanted to isolate a low abundant protein, PSA, for example, you could put it there, capture it, and put it through the automation system. So lots of possibilities there. So the system's out there. We love it. And people are starting to pick it up. It was transformative for our lab to move everything to an automation system.
Yeah. So I have just a quick comment there, Jenny. So I saw you have a 4-hour trypsin digestion. There might be a chance to reduce that to speed up your assay even further.
Yeah, so that would be super cool. We should collaborate on that because I think it would be really cool to be able to increase it. And I think there's chemistries that you can do to do that as well, right?
Right. We're also looking to use TCAN as a digestion.
So all of these automation platforms will work. So TCAN will work. I mean, all of them. What you want is you want to have something that's a robust platform, right? So there's a lot of really, these automation systems, that's their business, right? I mean, and so you want to work with them and get it in and work, through everything. So our, probably the thing that I would say is the most important was our incubator is humidity controlled. So we don't lose the evaporation. and then getting the salting on, which is a big place you get a lot of CV issues, is the salting, and having that on the automation system has helped a lot.
Yeah, thank you. So Doug, you want to go for the next
Yes,
question?
I think we're down to 2 minutes. Maybe we can do one more question. There was a question in here on, did you study carryover for your workflow, and did you see differences with the various steps in your workflow? Can you speak to carryover?
Yeah, so So Chin-Fu, who has led our automation, is just a brilliant scientist. And so when we were building it, we did things like, let's spike in 10, 15 label peptides into every buffer that we're going to put through the automation system, and actually then do MRM at the back end to find out what's happening through everything. So yeah, we've done that across the board. But I will address two where you have to be concerned. One is the depletion, but those beads we don't reuse, which is really unfortunate. So there has to be cost effectiveness. We will want to be able to see whether or not you can do that, right? Because when we years ago worked a lot on this, probably 10 years ago, and there are ways to regenerate, and you just have to figure out that cost benefit, where that cutoff is. But right now what we do is we only do a single depletion, so there's no carryover there. On the mass spectrometers itself, both the EvoSept system versus Phenomnex system. So the EvoSept, you have to probably be double check it for yourselves. They're single unit for us, they're throwaway, so we don't, but it can be a problem. And then Phenomnex, our wash system, we spend a lot of time tracking carryover on it to make sure that the wash cycle, the cycle that you do to regeneration, and we actually monitor that over time. Little bit crazy, but yeah, so it's absolutely super important, especially for low quantity analytes, right? I mean, this is very important. Great question.
I think that covers questions, and I think we're right at time. Jenny, excellent presentation, great discussion, really appreciate your contribution. Any last words?
Yeah, thank you so much, Jenny. It's a really good topic on public health. It's very interesting. Of course, I will ask questions. I will follow up with you.
Absolutely.
So for audience, I really encourage you to attend the technical seminars. We really appreciate all the vendors sponsoring this event, and please go visit them and talk with them to see what's the latest technology we can take advantage for our everyday life, everyday job. Thank you so much for your attention. Thank you, Jenny, again.
My pleasure.
Thank you. Thanks, everyone.
Thank you.
Bye.